we are back to the French accent. I don't know, but I think I'm in between Philippe and Rodolphe with regard to the quality of the French accent. So it's my great pleasure to be here at the University of Toronto. And surely I would like to thank the Gairdner Foundation for sharing the prize with my colleagues and for having rewarded the CRISPR-Cas9 field. And I would like to say that I was very happy the last 3-4 days to really give a number of talks in Montreal and in Toronto, also in front of high school students. I think it was very much enjoyable and actually a reason to reflect a little bit on science and what it is to be a scientist. And also the reflection, which is that actually there is nothing better when you're a scientist to just develop a story that can have an impact, and this has been actually a kind of obsession, is to try to have a story. So focus on the quality, not always on the quantity. So I will start with this slide because this has been the focus of my research over the years, the understanding of diseases at the fundamental level. And actually, so I was told to try to use the mouse, so I will try to use the mouse. Actually, the main focus of my research has really been to focus on bacterial infectious diseases among different other types of infectious diseases which my colleagues are focusing on. Others are focusing on other types of diseases, but like everyone who's interested in diseases, we all want to understand mechanisms at the cellular and molecular level. And we always hope to identify pathways, genes, and molecules that would allow really to understand the cells and organisms we study and to understand how certain pathogens, in my case, bacteria, really cause diseases. And also with a hope to develop novel therapies and novel technologies, and this is what CRISPR-Cas9 is about with regard to the technologies. And I'm very glad because there is William Nava, professor at the University of Toronto, an old colleague of mine, and he knows that when we were struggling as postdocs at the New York University Medical Center, people were wondering why I had an obsession on genetic tools. And as a matter of fact, I developed a genetic tool to facilitate really the genetics and allow precise genetics in bacteria, in gram-positive pathogens. And also along the way, I was focusing on transgenic mouse models of skin diseases, also with the idea that it was important for me if I wanted to develop my own niche in bacterial infectious diseases, but enough to understand that genetics in mice was not so easy, first of all, and that second of all, the best was really to work with human cells and human tissues, and I'm very happy that the CRISPR-Cas9 technology allows this new perspective in my field of infectious diseases. So you know the CRISPR-Cas system with the 3 phases, spacer acquisition, the immunization, memorization phase, okay. Then the expression of the RNA components and protein components, and then the interference system. And Rodolphe and Philippe explain you well the different steps in the history of the CRISPR-Cas biology, and also in the history of the certain aspects of the applications, and also the fact that this CRISPR-Cas system has largely evolved into 6 types. I mean, this was the latest nomenclature published in 2015 with the CRISPR-Cas9 system that is the first minimal system that has been studied, so the type 2 system, with obviously a different setting than the type 1 and the type 3 systems, whereby the dogma for the type 1 and the type 3 systems were really that one needs the CRISPR-associated proteins encoded by the Cas operon, and the precursor CRISPR-RNA molecule encoded by the CRISPR array that will undergo maturation, and then a complex of CRISPR-associated proteins guided by these mature CRISPR RNAs to target the invading genome. And also having in mind that the repeats would form hairpin structures thanks to palindromic sequences, but that in principle this was self-sufficient. You needed the proteins and the mature CRISPR RNAs. So we all know that for the type 2 system it is different, and this is what I'm going to explain you briefly, is how we came to the understanding of the CRISPR-Cas9 pathway. So this is about this technology that is a two-component system in the sense that you have the Cas9 protein and the RNA component. The RNA component in nature are two RNAs, CRISPR RNAs and tracrRNA, that can form a duplex and that are linked together by a linker to lead to the single-guide RNA molecule. But depending on the way to deliver the systems, either the two RNAs or the single-guide RNAs are used. And this RNA component guides Cas9 to the site on the DNA to be modified thanks to the base pairing of the green sequence of the CRISPR RNA and the corresponding sequence on the DNA. There is one element, and you've heard about the PAM sequence, that has to be present downstream of the piece of DNA that is targeted by the system. And in the case of Streptococcus pyogenes, that is really the system that is used worldwide for genome editing in cells and organisms. This is the PAM for Streptococcus pyogenes is NGG, so a short stretch of nucleotides which one can find relatively easily on the genome. And this is critical for the system to work. So it's really a programmable system whereby only the green sequence of the RNA has to be exchanged, and biologists can use this system to really perform precise gene surgery and also change expression of genes in a targeted fashion. So the system originates from Streptococcus pyogenes, at least the system used for the genetic tool. And just to show you here the Streptococcus pyogenes, so you can see the cocci that are colored in red for the purpose of the picture, invading epithelial cells, colored in green for the purpose of the picture. So these are human pharyngeal cells. And this pathogen is responsible for a wide range of diseases, from mild infections such as pharyngitis to more severe infections such as necrotizing fasciitis and streptococcal toxic shock syndrome. So group A Streptococcus can really cause this severe invasive infection thanks to a battery of virulence factors, some of which are encoded by lysogenic phages. And this is also one of the reasons I got interested in pursuing on the CRISPR research, and you will understand why later. So in the laboratory, we try to focus on different types of mechanisms, focusing on the regulation of expression of genes at the level of post-transcriptional, translational, and post-translational. And we try to understand how bacteria can cause diseases, can attack the human host, and also to a certain extent how the bacteria can adapt and survive in the human host. And we are also interested in how the human host defends itself against the bacteria. So we focus on regulatory mechanisms involving proteins and also small RNAs, with really trying to understand how all the different factors, like, for example, virulence factors involved in really causing diseases, all those regulatory mechanisms can work, taking into account that the bacteria are always under very strong stress in their environment. So about now maybe 14, 15 years ago, I started to be interested in small RNAs in Streptococcus pyogenes, having in mind that small RNAs in bacteria can interact with messenger RNA via antisense mechanisms, changing the stability of messenger RNA or having an effect on the activation or inhibition of initiation of translation. small RNAs can also interact directly with the proteins and activate proteins or sequester them from their functions. What was missing at the time, if one wanted really to have a new mechanism, knowing that you have a large variety of diversities of mechanisms in the different interactions which I have just discussed, the fact that a small regulatory RNA could change the regulation of another small regulatory RNA, and the fact that a small regulatory RNA could interact directly with DNA. So at the time when we started to work on this class of molecules, this was not described yet, and this is really what the CRISPR-Cas9 system brings. So about 10 years ago, we wanted to know more about small RNAs. We were focusing on some for which we could show that they had a role in the regulation of virulence factors in Streptococcus pyogenes, and we really performed a bioinformatic analysis, and here are the small regulatory RNAs for which we could really show a very nice expression by Northern blot analysis. We picked some of the RNAs, including tracrRNA and also CRISPR-RNAs. And coming to CRISPR-RNAs, in Streptococcus pyogenes, like in other types of bacteria, this system, as you know, is really a defense system that allow bacteria to resist the invasion of mobile genetic elements, plasmids, transposons, and phages. So in Streptococcus thermophilus, you've heard that the CRISPR system is really allowing the bacteria to defend themselves against death. So this, we really talk about phages that enter the lytic cycle and that can produce, that can kill really the bacterial cell and propagate to kill other bacterial cells. In the case of Streptococcus pyogenes, the CRISPR systems are composed of spacers that can target lysogenic phages, temperate phages that enter the lysogenic cycle, whereby the invading DNA of the phage can integrate into the genome of the bacterial cell and reside as a prophage and bring new genes. In the case of Streptococcus pyogenes, those genes encode virulence factors that are important for the severe forms of diseases caused by Streptococcus pyogenes. So coming back to tracrRNA and CRISPR-RNA, we picked up tracrRNA because we showed that it was well expressed by Northern blot analysis. This is expression of total RNA from Streptococcus pyogenes grown from early log to late stationary phase and probed with tracrRNA, so at the time called spRNA-49. So you see really the three forms of the RNA. We could map at the time via primer extension and race analysis the 5' end and the 3' end of the RNA to figure out that really this RNA was encoded in the vicinity of a gene that was encoded, that was annotated at the time to be a gene encoding a CRISPR system-related protein containing the RUC-like nuclease domain and the HNH-like nuclease domain from S. thermophilus. So this is now what we know is a Cas9 gene that had, as it was explained, different names, CSN1, Cas5, and CSX12 in the past. And this tracrRNA that is really constitutively expressed and can undergo maturation with a processed form that is corresponding to the third form that you can see on the Northern blot. However, at the time, we were really interested in finding messenger RNA targets, like a lot of biologists interested in small RNAs. And we found the messenger RNA target encoding a virulence factor that can lyse blood cells, so it's an extracellular protein involved in erythrocyte lysis. We really knocked out tracrRNA, but we could not make any sense of this interaction in the sense that we could not find, looking at the expression of this virulence factor and also the different phenotypes associated to this virulence factor, we could not find any role of tracrRNA in a change of expression of this virulence factor. So now we know why, and it's a bit complicated to explain, but we were a bit desperate, and we wanted really to find a function for this RNA. And the idea that I had was maybe that tracrRNA has actually a role in the type 2 system. The reason why is because you have a lot of RNAs in bacteria that are located in cis and can act in cis, or located in cis and can act in trans, or located in trans can act in cis, and so on. And that actually at the time we didn't know anything about the regulation or the activation of the systems. So we were lucky enough to have the type 2 system in Streptococcus pyogenes. That is really the minimal system with only 4 genes in the operon. And the next slide is really to show you this interaction between tracrRNA and the CRISPR-Cas system, having in mind as well that we were interested and we were starting to work on the CRISPR system to show a role in the acquisition of virulence genes via the acquisition of temperate phages. So this is it's an experiment which we performed in 2009 in the lab, and that shows pretty much everything of how one can understand really this particularity of this enzyme guided by 2 RNAs. So on the left side, you see the expression of tracrRNA with 2 promoters driving the expression of 2 primary forms of the RNA and a processed form. You see here by Northern blot analysis in the wild-type Streptococcus pyogenes is processed form. If you knock out CRISPR-RNA, you lose the processed form. If you complement the knockout of CRISPR-RNA with CRISPR-RNA in trans, you rescue the processed form. And on the right side, you see the expression of the precursor CRISPR-RNA that undergoes the processing, giving rise to the mature forms that are composed of a short portion of spacer in 5' and a short portion of repeats in 3', whereby here you can see really the mature forms of CRISPR-RNAs. If you knock out tracrRNA, you lose the mature form of CRISPR-RNAs, and if you complement the tracrRNA knockout with either primary form of tracrRNA, you rescue the processing. So this was really an indication that those 2 RNAs are co-processed, and it was clear at the time that they will be co-processed via an interaction, and that is logical when you deal with 2 RNAs. Having said this, we were also starting a collaboration with a group of Jörg Vogel to perform what we call a differential RNA sequencing analysis in vivo that would really allow to identify in vivo RNA species and also identify in vivo their 5' and 3' ends. So it was clear as well that there was a first cleavage that was occurring within the repeats, like for the type 1 and the type 3 systems. However, for the type 2 systems, those repeats do not have palindromic sequences, so they cannot form an hairpin structure. And this somehow intramolecular hairpin structure that is formed by the repeats of type 1 and type 3 are here replaced by an intermolecular hairpin structure between the repeats of CRISPR-RNA. Here you have the spacer that can target the phage genome, and the anti-repeat of tracrRNA. So when we looked at this, when we showed that there was really this interaction between the 2 RNAs in the lab, the first thing was really to look at the in vivo RNA sequencing analysis, which we were analyzing in my lab, and figuring out that the cleavage was occurring inside at the level of the anti-repeat-repeat interaction, and was giving that this cleavage was giving rise to short 3' overhangs that are typical for cleavage by RNase III-like enzymes, which is reminiscent to processing of macroRNAs and small interfering RNAs via RNase III-like enzymes like Dicer and Rocha. So we thought that for the evolutionary point of view, it was a very interesting observation. But more than this, what we were really puzzled with is that in contrast to type 1 and type 3, whereby there is really a CRISPR-associated protein that is involved in the maturation of the CRISPR, the precursor CRISPR-RNA molecule, here we had two additional elements, another RNA molecule, tracrRNA, and also the RNase III from the bacterium that really takes care of this first processing event. So this is really an unusual pathway for RNA maturation, and we really used actually, as a matter of fact, the tools that I had developed in the past to really do very precise genetics on Streptococcus pyogenes to have a genetic strategy combined with a biochemical strategy that showed the following. tracrRNA forms a duplex with the repeat of the precursor CRISPR-RNA, stabilized by the protein Cas9, at the time called CSN1. This duplex is cleaved by RNase III. It gives an intermediate form of CRISPR-RNA with repeat in a portion of the repeat in 5', full spacers, and then a portion of the repeat in 3', forming this duplex with tracrRNA. and then a further trimming, which initially we thought that Cas9 may be involved in this maturation step, but we could not show this in vitro. And we think that there is another endoribonuclease or exoribonuclease from Streptococcus pyogenes, which we could not identify yet, that is responsible for the further trimming with protection of the mature form of CRISPR-RNAs with Cas9 to really lead to this mature form of CRISPR-RNA still bound to tracrRNA. So there was for sure the question whether tracrRNA was only necessary for the first step of maturation or whether it will leave the system. But with certain experiments which we did to really look at the conservation and the diversity of the system, we had means to believe that this was not the dead end for tracrRNA and that tracrRNA CRISPR-RNA interaction was forming the kind of anchor to bring Cas9 to the invading genome. But we needed to show this, and before we continued with the biochemistry, we were interested in showing that the system of Streptococcus pyogenes was active. So very early on, we saw that the spacers of the system were targeting lysogenic phages. So this was already described in the literature, but more in the prospect of using these additional spacers as a way to type further the clinical isolates that are classified by M serotypes. But here we wanted really to show that indeed the system was active, biologically speaking. So we developed a readout system using plasmids in which we cloned a phage sequence that can be targeted by a spacer of the CRISPR-Cas system. And we showed that if we deal with a wild-type bacterial strain, and if you have the phage DNA entering into the bacterial cell, the CRISPR-Cas system will recognize the phage DNA, and this will be the dead end for the phage. And this will be translated for the biology of Streptococcus pyogenes into no acquisition of virulence genes. And if you knock out CRISPR-RNA, tracrRNA, the gene encoding Cas9 and the gene encoding RNase III, you will lose the immunity, whereby the system is not active any longer, and the phage DNA can insert itself into the genome of the bacteria, so you have acquisition of prophages and therefore acquisition of virulence genes. So this was to show that CRISPR-Cas system has a spin in virulence fitness by limiting the acquisition of virulence genes. Then the next step, and this is another experiment that is really said again pretty much everything for the next step, was really to do biochemistry, whereby the idea was really to purify the Cas9 from Streptococcus pyogenes, and then use a plasmid in which we cloned the phage DNA that can be targeted, then the CRISPR-RNA, the tracrRNA, which you can see here forming a base pairing with a repeat. So the CRISPR-RNA here has a green sequence, the spacer that can base pair the sequence to be targeted in the presence of magnesium. We did this assay on a plasmid circular and linearized, and what we could show is that Cas9, guided by the two RNAs, cleaves the target DNA 3 base pairs upstream of the PAM, which is corresponding to the data which Sylvain Moineau had in his paper that he published in 2010, where he had described prior to this publication the biology of the PAM and the fact that the PAM was important to discriminate self between non-self, but also showing that in vivo the system could also trigger this cleavage 3 base pairs upstream of the PAM. However, at the time, the tracrRNA was not identified, and the system was not studied at the genetic and biochemical level to understand the mechanism. So what Cas9 is doing is that Cas9 has a domain that allows it to first scan the genome to find the PAM. Then there is binding to the PAM, and then homology search with the spacer sequence of the CRISPR-RNA to find the sequence that can base pair with this green sequence. You have always, so this triggers R-loop formation, always a change of conformation of the protein after it binds the 2 RNAs, after it binds the PAM, and after you have strand invasion to position the 2 nuclease domains so that HNH can cleave the strand homologous to the target DNA, so forming a base pairing to the target DNA, and the RUFC motif cleaving the strand that is not complementary to the CRISPR RNA with a double-stranded DNA break that is triggered. So again, those two domains that were initially predicted by bioinformaticians. And so this idea that one can simplify the system and have a single guide RNA instead of two RNAs, as it was explained by Jennifer Doudna, and if you introduce a double-stranded DNA break in the DNA in a targeted fashion, you do exactly what zinc finger nucleases and TALEN nucleases are doing. There are also sequence-specific nucleases that were used prior to CRISPR-Cas9 and are still used by some biologists. They are more complex to engineer because the code for recognition of the DNA to be modified is contained within the protein, so one has always to somehow engineer a new protein for a new piece on the DNA to be modified. So if you introduce a double-stranded DNA break, you trigger a non-homologous end-joining repair mechanism in higher organisms that will lead to larger or smaller deletions at the site of the break. You can also introduce an homology donor with homology arms upstream and downstream of the break and replace a sequence of interest by another sequence of interest, and you can introduce mutations, correct mutations, or introduce new types of sequences. And here, really this hypothesis that the system would really be the simplest system, this was clear working on type 2, that with one protein involved instead of a complex of proteins, this will be the system the most amenable for genetics, and with really this proposal that it could really offer considerable potential when one compares the system with zinc finger nucleases and TALEN nucleases for genome editing in cells of the three kingdoms of life for biotechnological, biomedical, and gene therapeutic purposes. And as a matter of fact, 6 to 8 months after we published with Jennifer Doudna the science paper in the summer 2012, so 6 to 8 months later, it was shown by different scientists that the system was working very well in human cells, and Feng Zhang will explain you what he has done with regard to the development of the technology in human cells, that it was working to perform knockouts in mice and multiple knockouts, that it was working in plants, in zebrafish, in yeast, in flies, in worms, so in a lot of organisms that are used as model organisms to understand mechanisms of life, and that later on was shown to be useful for organoids and also monkeys. Everyone agrees to say that the technology is very cheap, it's easy to use, it is very efficient, specifically the system from Streptococcus pyogenes. It's very versatile, it allows multiplexing, whereby one can really design different guide RNAs to target different sites on the genome. For a natural system, it's already highly specific, and you have a lot of improvements with regard to the specificity to limit the off-target effects. That is, that the system will recognize other sequences on the genome that it is supposed to recognize by the RNA. It has low toxicity, and a lot of efforts are done with regard to delivery purposes that are a little bit the bottleneck when one also considers that now the tool offers the possibility, as Jennifer said, to manipulate organisms that were not easy to manipulate prior to CRISPR-Cas9, where surely delivery tools are also missing in this matter, and for everything that is human gene therapy, a lot of efforts in this direction. Some aspects with regard to the Cas9 toolbox. Cas9 can, as I said, can really trigger the generation of small or larger deletions, rearrangements on the chromosome, insertion of sequences or replacement of sequences, introduction of mutations, and also correcting mutations. Now there is a very nice feature with regard to Cas9. It has, as I said, 2 nuclease domains in nature. And the idea, and this was also tested by the group of sickness, was to take the opportunity that there are those 2 nuclease domains to knock out one domain and transform the nuclease into a nicase, or knock out the 2 nuclease domains and transform the RNA-programmable nuclease into an RNA-programmable DNA-binding protein that could actually inhibit transcription if one targets the protein to promoter regions, but one can fuse to this dead Cas9 different effector domains, such as domains that can activate transcription, inhibit transcription, domains that can change the epigenetic marks on the chromosome, or domains that can allow to label site-specifically the DNA and can really have this kind of toolbox that allows a lot of tricks on the DNA, not only for really changing the sequence of the DNA, but also changing the expression of the DNA and labeling the DNA or modifying the DNA. And Jonathan Weissman tomorrow will explain you all the screens that he has developed in this regard, so CRISPR activation and CRISPR inhibition. So you have a lot of developments going on also to use the technology for additional purposes. It was already said that the technology really has large applications and allows also to consider a number of fields of research in a progressive manner and really in a transformable manner, such as in agriculture for the production of crops. Also an impact on the field of synthetic biology with the engineering of pathways. Also in the field of gene drums and human medicine, whereby the technology is very useful in human medicine, at least indirectly prior to the hope that the technology will be useful in a more direct fashion. Indirectly because it really simplifies the genetics in human cells and humanized models, whereby one can really boost the understanding of functions of genes and unravel new pathways. One can also think of disease models in a more clinically relevant manner, so really developing these humanized models of diseases. There are lots of efforts to use the technology to really improve and develop the screens for new targets for therapeutics, also to validate new therapeutic targets and medicine under development. A very nice approach with the efforts to sequence the human genomes, at least the genomes of patients, whereby more and more genetic predispositions are associated to diseases. So it's really the bed-to-bench approach, going back to the bench and test those genetic predispositions. And surely the challenge that is really to develop the technology as a direct therapeutic for human gene therapy, whether one is using an ex vivo approach combining gene and cell therapy or an in vivo approach. I will talk briefly about the evolution of the system. So it was said at the beginning of the morning that the system has largely evolved. And what is really clear when you work on one type is that actually you have a large diversity of sequences. And this is really something that puzzled us at the beginning and which allowed us also to really understand the mechanism, is that we were identifying this tracrRNA, and the first question that we ask when we have a small RNA in bacteria, it's I think what every biologist will do when one identifies a new molecule, is whether this molecule is ubiquitous or whether this molecule is really specific to the species with which we work. And when we tried to blast this tracrRNA, it was looking like nothing. So we ended up with the hypothesis that this tracrRNA is really specific to S. pyogenes. That is why we were also following up with a messenger RNA target, until we understood that tracrRNA, the characteristic of tracrRNA is to form an anti-repeat interactions with the precursor form of CRISPR-RNA. And having in mind that even if you look at the type 2 system in different bacterial species, you will always have a diversity of sequences of the repeats. You will also have a very high diversity of the sequences encoding the Cas proteins, like the Cas9 protein has very, very low amino acid identity with only conserved domains with regard to the domains that can be involved in the strand invasion of the CRISPR-RNA upon R-loop formation, and also the domains cleaving the DNA. Otherwise, it's extremely diverse. The Cas9 proteins have also different sizes in nature when you look at the system in different bacteria, and we did a bioinformatic analysis that we started pretty much early on, where we could find the system in more than 350 bacterial species with a large diversity of sequence. So this tracrRNA has always an anti-repeat sequence, but the anti-repeat sequence is always of different nature, and then upstream and downstream of this anti-repeat sequence, the sequences are very variable. But this allowed us to understand very early on that there is a conservation of this dual RNA structure, even though the sequences of repeat and anti-repeat is different. And if you classify Cas9 via phylogenetic groups, so you can have some Cas9 belonging to the same phylogenetic groups, and they correspond to RNAs that have, in a way, a certain structures that are typical for the same phylogenetic group, and also some interchangeability, whereby Cas9 proteins from one bacterial species could potentially function with a dual RNA from another species belonging to the same phylogenetic group, but will not work with the dual RNAs of a bacterial species belonging to another phylogenetic group. Having said this, you have also a large variety of the PAM sequences. Everyone is happy with tracrRNA because it's one of the systems that maybe the system that is the most active as a tool for genetics in eukaryotes, and it has this NGG PAM that is not so restrictive. But when you look at other bacterial species, and this was said, you have always a variety of PAM sequences. So the idea for us was really also to look for smaller versions of CRISPR-Cas9 that are more amenable for genetic tool purposes, longer version of Cas9, try to find what will be different in the domains and try to work with different chimeric forms of the proteins and the RNAs to enhance this toolbox. Having said this, there is other systems that are minimal, like the type 5 and type 6 systems that have been studied very recently, and I just want to show you an example because before, with the connection between tracrRNA and the type 2 systems, I actually was thinking that maybe Cas9 will be doing what Cas6 and the complex of proteins do for the type 1 and type 3 systems, meaning it will do everything, the maturation and the targeting of the DNA. And this is indeed what the protein does. So we worked on this protein because we were interested in the long Cas9 from Francisella novicida, that is another bacterial species causing diseases in humans. And in this bacterial species, you have two systems, type 2 and type 5. And we had the RNA-seq analysis of this CPF1 system, the type 5 system, where we realized that there are hairpin structures formed by within the repeats of the long precursor CRISPR-RNA molecule, and that the production of the mature RNAs were repeat in 5' and spacer in 3', where we could show, using mainly a biochemical analysis, is that CPF1 contains two nuclease domains, an RNase motif and a DNase motif. CPF1 can recognize the repeats of the long precursor CRISPR-RNA molecule, cleave at the level of the repeats, and then use CPF1 can use the mature RNA that it has processed to be guided to the target DNA and cleave the DNA in a different fashion, not introducing a double-stranded DNA break in a blunt fashion, but producing DNA species with 5 nucleotide 5' overhangs. So it's really a neat example in nature of a protein that has really a specificity for a DNase and an RNase function. So the group of Jennifer Donna and Fang Zhang showed recently that C2 can use the same principle, but C2 can process the RNA, and in this case, it does not target DNA, but targets mRNA. So as you have heard, this field of research involves a lot of scientists that have worked in the past, and they are still working, asking a lot of interesting questions on new systems of CRISPR-Cas, but also this step of spacer acquisition and adaptation. You have very nice work done in this area. And I really do hope as well that with the effort of the community working on bacteria and bacteriophages, that we will discover in the future additional defense systems that could be harnessed as genetic tools, always having in mind that one should not forget that molecular biology has really started with macrobiologists trying to understand how bacteria defend themselves against bacteriophages, and also macrobiologists working on viruses. And all this research has really led to all the enzymes which we know are used in molecular biology in nowadays, restriction enzymes and so on. So I would like to thank briefly the different institutions where I performed the work in Austria, the University of Vienna, Max F. Perutz Laboratories, Then the main work was developed during my time in Sweden. So the Laboratory for Molecular Infection Medicine, Sweden at Umeå University in the North Pole, nearly, an affiliation with EMBL of Heidelberg. Then I moved 4 years ago at the Helmholtz Center for Infection Research in Braunschweig, Hannover Medical School, and I moved again a year ago at the Max Planck Institute for Infection Biology in Berlin and Humboldt University. This is part of my team. I would like to thank from my lab Krzysztof Szylinski and Elica Delcheva, who really did a wonderful job. Elica Delcheva, master student, Krzysztof Szylinski, Ph.D. student. Other scientists who have contributed to the work, essentially also Inès Fonfara, Anaïs Lerin. Other scientists who focused on the CPF1 story lately, Inès Fonfara, Hagen Richter, Maida Bratovic, Anaïs Lerin. My collaborators, a group of Jörg Vogel with Cynthia Sharma for the step of maturation of the CRISPR-RNAs, the group of Jennifer Dana for DNA interference, and she mentioned Martin Jinek, who has now his own lab at the University of Zurich, and the bioinformatician, the group of Eugenie Konin with Kara Makarova for the evolution part. The two companies which I have to cite for conflict of interest, co-founded with Rutger Novak and Sean Foy. And this quote from François Jacob, a French microbiologist: nature is a tinkerer, not an inventor, and it's all about bricolage in nature and how we can harness this bricolage to make better bricolage in an engineering fashion. Thank you for your attention.